Thank you. Last day, last lecture. All right. So I worked out Turing patterns and Turing conditions yesterday. There was some question about the determinant. Did that sort out? Should I work it out? If you do, trust me, if you do the algebra, you'll just get back one of the conditions that I had derived earlier, nothing more than that. Okay. So then I'll move ahead, and I want to show you some aspects of mechanics, how mechanochemical patterns come up in living systems, and in line with the spirit of this school, how geometry comes in. Okay, so the classic paradigm for understanding pattern formation is reaction diffusion equations, the kind that I showed you earlier. These kinds of patterns are actually pretty useful even in living systems. For instance, many creatures use simple degradation, diffusion, coupled with some boundary conditions to lay out the patterns. In the Drosophila wing disk, turns out the anterior-posterior patterning, the front-back patterning of the wing disk is actually accomplished by a certain pattern in the DPP molecule, decapentapegic molecule. So now it's not difficult for you to see from here that one can construct something which is a periodic stripy pattern. So here's the idea. Let's say there's one molecule A which has this exponential profile like this C. And then you say that A interacts with B and C in the sense that A makes B and C. So the arrow indicates a positive interaction. But A inhibits D. So then you can see if there's a profile of A which is set up by this boundary condition, clearly B and C will follow the profile of A, but D will be antagonistic to A. Well, complicate the thing, C and D make E, B inhibits C, B makes... So at the end of the day, you can cook up something which will give you a very nice periodic stripy pattern. And those are the kind of stripy patterns that are actually seen in the fly. Okay, so these are not just abstract molecules. You can actually find those molecules. The initial one which sets up this exponential profile is becoid. And then there are patterns which come up with hunchback, rupal, giant, and so on and so forth. At the end of the day, you get these very nice, colorful, stripy patterns. Those stripy patterns are actually what lay out the body plans of the creature eventually.
So just to clarify, here they don't come from an instability. So it's a different...
This is not an instability, this is just a boundary-driven pattern.
Yes, and you need one different species per pattern?
Yeah, so you pattern one of them, let's say A, driven by a boundary condition like so. And the boundary condition is actually set up by, in the case of the fly embryo, the mother deposits some maternal mRNA in one corner to make one of the proteins. So there's like an extra source of a molecule there.
Yes.
And the rest of the thing is just logical chemical reactions.
In the two last rows, so the first three lines, there is one peak per species?
This is not species. This is one peak per stripe. The stripes that I showed you earlier, which lay out the body plans in the creature.
No, but on the left, there are chemical reactions.
Yeah. These ones, yes.
Because on the two bottom, there are FG, SSS.
So don't make a one-to-one correspondence. This is just an illustration that if I give you enough complexity, I can cook up any periodicity. But here are the real ones that make the periodicity. Okay, so the Turing patterns that I showed you actually result from an instability. And so like I showed you yesterday, you start with something which has diffusion and reaction. Reaction changes numbers, diffusion transports and mixes of particles. So like I showed you, you need to have the following conditions, that the molecule A, in this case, yesterday I think it was phi1, is an activator in the sense that it makes more of itself. So you indicate that with the arrow. The molecule B is the inhibitor, so it tries to reduce its own concentration. If that is the case, then the diffusion coefficient of A must be less than the diffusion coefficient of B. If so, the homogeneous state is spontaneously unstable to fluctuations. So here's a chimograph from a numerical simulation. You start from something which is more or less uniform. I'll show a video in a minute. And as you progress in time, the periodic patterns spontaneously emerge as an instability of the homogeneous state. So if you worked out the math yesterday, you would have noticed that there is a bit of an ambiguity. The connections between A and B, whether A activates B and B inhibits A, or where A inhibits B and B activates A, is left unresolved. So what you get is just the product of A12 and A21 should be positive or negative, I don't remember. But it doesn't say which one. So there are two classes. One is called an activator-inhibitor system. The other is called activator-substrate depletion model. So here's an example, Geirer-Meinhardt model. There are plenty of models people have constructed to explain Turing patterns. So here's one more brusellator. This was discovered in Brussels, and it shows oscillation, so brusellator. If you actually run the simulation, it's a numerical simulation of these PDEs, start with a simple homogeneous state with small random fluctuations. the fluctuations get amplified and the homogeneous state destabilizes, and you spontaneously get the appearance of periodic patterns. So remember the symmetry breaking that I talked to you yesterday? Start from something like this, but this state is unstable to fluctuations, and what you get is something which is periodic in space. Now, this is all nice, this is all theory. Does it actually work? Like I said, it was difficult to find out that Turing systems exist in real life for a long time. Chemical systems were one of the first designed to test these Turing instability, and they worked. But in real biological systems, there was a lot of speculation. It should be fish, not Fisk. There are a lot of speculation that many biological patterns are actually Turing patterns. Classic examples being fish stripe patterns. So this is one of the first papers which actually showed that there is a reaction diffusion wave on the fish stripe patterns. This is still qualitative. One of the papers the follow-up paper by the same group did something really nice. Fish stripe patterns, very nice patterns that you see. The way they probed the system was to take this and do a laser ablation, mild, so you don't hurt the creature. And then you just follow that in time. So the creature is growing, this is a juvenile. And as it grows, you see these stripe patterns, but not only that, you develop this bump. Okay, so that's typically something that you see in numerical simulations of Turing patterns. So here's the idea. So that's the experiment. I don't know if this is visible. Is it visible? Probably not. Can we dim the lights? Dim the lights. So yeah, good, thanks. It's very faint, but you can see a Turing pattern like stripy Turing pattern that has been set up. And then just like to mimic the experimental perturbations, you just remove the concentration here, and then you ask, if I just go through the reaction diffusion dynamics, what's the kind of pattern? You see something like a hump coming up there from the third thing. So this was qualitative evidence that really pointed out that Turing patterns possibly lie biological systems. Mammalian digitation patterns, like I mentioned to you, very good proof actually came up just about 10 years ago, where people showed that you can even control the genes, which will eventually make the proteins and their properties, the chemical reactions, the diffusion constants, and eventually those are the ones that are actually involved in patterning. So here's a more complicated thing, just like your hand, you think about this, the size of the system is changing as you go ahead. So in the sense you have something which is thinner here and then widens out. And what you want is fingers to start somewhere at the base and then to go up and then to stop somewhere. So it turns out in mammals, in the mice, this is controlled by a morphogen gradient, like the concentration field that I showed you earlier, with a boundary condition pattern on this side and a Turing pattern on the transverse direction. This paper is something I recommend. It has a huge 100-page supplement with all kinds of detailed reactions, and people show that Turing patterns really are underlying mammalian digitation patterns. All right, so there are Turing patterns and Turing patterns. So you can, make the French flag and the British flag and the US flag and whatever flag you want. All this is nice. The Indian flag is a little difficult. I've tried. It's not that easy. But this is a conceptual picture that is missing here. These are all reaction diffusion patterns, in the sense that the change in the chemical concentrations happens because of random walks, diffusion, and chemical reactions. There's no mechanics. Right? There's no forces, there's no stresses coming into the game. Now, if you watch a movie of development, it's impossible that there's no mechanics, because now, as you see the formation of structure, it's not just chemicals coming and attaching and you having chemical reactions coupled with diffusion, but there's very clearly shape changes, geometry changes, and the very fact that you're molding the system into shape, immediately it should tell you that there are stress patterns, there are mechanical patterns. So where are the mechanical patterns and how are they talking to the biochemical signaling patterns, the gene patterns? And so therefore there must be a crosstalk between biochemistry or genetics and shape, the emergent form, and the crosstalk must be mediated by mechanical forces. So where are the mechanical forces in living systems?
So Vijay, I had a quick question. So even in the Turing type system, is there feedback in the manner that the diffusion coefficients and all the constants are dynamic in some sense?
So it turns out for this system, they took the growth from the experiment, put it by hand, and then solved a diffusion equation on a system where the growth is prescribed by hand. So there was no feedback. It's just a reaction diffusion system on a deforming domain, where the deformation you prescribe by hand. I'll show you something where the deformation can come up spontaneously. Yeah, so there must be mechanics. And so where is mechanics? This is not new. I mean, people have thought about this from 100 years. If you don't know, D'Arcy Thompson is something that every scientist should read, without a doubt. And way back, even before quantum mechanics, he sort of postulates the laws of physics and chemistry must underlie living forms. And the kind of insight that he gives is remarkable. So the central question is, there are two parts. One is, where do you get mechanical forces in cells and tissues? And what are the principles behind mechanochemical patterning? So that's the kind of picture that I work on. I look at the intersection between geometry, mechanics, and biochemistry. I'm not a biochemist. Biochemistry for me is just nonlinear chemical reactions. The primary structure that generates mechanical forces in all eukaryotic cells is what's called the cytoskeleton. Just like we have a skeleton, the cell also has a skeleton. And the cellular cytoskeleton has three main components. One of them is the microtubular cytoskeleton shown in green. The other is the actin cytoskeleton, which is predominantly on the periphery of the cell. It's like a cortex to the cell. And there's the nucleus. There's also another part which is called the intermediate filaments, which it's more of a cushioning thing, and one doesn't know much about what they do. So the cytoskeleton is a meshwork of filaments, motor proteins, cross-linkers, lots of gooey filaments. But the more important thing is that this is a system which is driven out of thermodynamic equilibrium by ATP hydrolysis. And the functions and the processes that happen in the cytoskeleton are responsible for moving cargo inside cells, but at a larger scale, even things like cell division and cell motility, cell polarization, and so on. So here are two examples. So cell division, like I showed you the movie earlier, is not just involving something where there's a copying of the genetic material, but there's also a segregation of the genetic material into two daughter cells. You can clearly see the segregation involves forces, pulling them apart. So the red is the DNA, histone. The green is the same green, the microtubules there. You can see the spindle-like structure that forms and pulls them apart, coming up there. In cell division, which if I have time, I'll show you some recent work, the actin cytoskeleton forms in the pattern of a ring, and the contractile forces in the ring are what lead to the constriction of the cell. So you can think of this like a balloon with a rubber band, which has been stretched and put there, and the rubber band will constrict the balloon into two daughter cells. So all these processes seem to happen spontaneously in a very self-organized and controlled manner. All right, so like I said yesterday, these are systems that broadly fall in the class of active materials. You can think about active materials as systems which are driven out of thermodynamic equilibrium. And in the case of living systems, the flux for driving you out of equilibrium is the lunch you had now. Because you have that, you're a non-equilibrium system, so therefore things can be run out of equilibrium. Examples again, the atomizing cortex, whether in living cells or reconstituted networks, wing discs, or even at a much larger scale, bird flocks, fish schools, insect swarms, and so on. So active hydrodynamics, you should really think about this as a formalism to couple chemistry, which is predominantly driven out of equilibrium, to mechanics, and therefore geometry as well. And so there is a very strong feedback between these two aspects. So I'll focus on the atomizing cortex. So the atomizing cortex, like I said, is a thin film, is a meshwork, a polymeric meshwork beneath the surface of the membrane. It's a very dynamic meshwork in the sense that there is a constant turnover of the material. So there is continuous polymerization of the monomers from the cytoplasm to the surface and depolymerization from the surface back into the bulk. So this sort of turns over the material. And so therefore on time scales which are longer than the turnover time scale, you cannot sustain elastic stresses because it will flow like a fluid. So this is essentially a viscoelastic gel. And so you can actually probe the Maxwell relaxation time constant by doing a laser ablation and finding out some recovery time scales. And this is roughly of the order of 5 to 10 seconds. The turnover time scale, depending on the cell and the state of the cell, is somewhere between 10 to 20 seconds. So if you're looking at processes that happen on much longer time scales, hundreds of seconds to minutes, you can basically assume the cortex flows like a fluid. So the cortex is throughout the periphery of the cell. So you look at whatever direction, you'll see a meshwork of polymeric filaments. But like I said, the thing that makes it active is the fact that there are myosin motors consuming ATP and tugging on the actin filaments. A coarse-grained description of this is the active stresses that I want to introduce. Okay, this is just a summary. I'll go through them one by one. So there's a vast literature on active hydrodynamics from the past 20, 25 years. What one does is to write down conservation laws for densities of conserved quantities, or if there are orientational order parameters, you write down equations for them. Typically, the systems that one looks at in living systems are at a very low Reynolds number, so you can typically forget inertia. So you do force and torque balance. So you write down equations for densities, concentrations of embedded chemical species. So for instance, this is where our Turing-like systems sit here. There's also a force balance, which if you use some constitutive relations will give you velocities or deformation rates. The constitutive relations can be something that come from viscoelasticity. In addition, you could have liquid crystalline orientational degrees of freedom, like polarity or nematic order parameters. Okay, so there's a bunch of review papers which sort of outline this formalism over the years. Okay, so I'll give a brief pictorial view of how to think about such systems. So this is a coarse-grained description, very definition hydrodynamics, is valid only on large length and long time scales, large compared to the molecular scales. The way you write down such a hydrodynamic description is going through what are called hydrodynamic variables. Typically, these are densities of conserved quantities or order parameters which break continuous symmetry. You assume local thermodynamic equilibrium. You typically write some sort of a linear response and look at some Onsager relations. And if you're not at equilibrium, you retain, you don't impose time reversal symmetry, you're permitted to break Onsager reciprocity. So for the cortex, like I said, this is a meshwork. This is about 0.5 micron thick beneath the plasma membrane. So on scales of the whole cell, 10 to 20 microns in size, this is almost a 2D film. So what you can do is to get the effective dynamics of the cortex by integrating out over the thickness direction and get effective two-dimensional dynamics for the active polar thin film. The cortex is to a very good extent an active polar thin film. Okay, so that's what makes the cortex an active material. People have speculated over a very long time that the cortex is actually responsible for many morphogenetic events, from cell division all the way up to sculpting organisms. Okay, so how does one write down equations? So look at rho, which is like the material density of the cortex. So you write down a constitutive relation, but like I said, there is turnover. So you can capture the turnover effectively by putting in a linear degradation rate. The velocity that's there for the advection part surely comes from some force balance. The force balance has stresses that come from the hydrodynamic stresses and the active stresses. I'll come to the active stresses in the next slide. At long time scales compared to the turnover, you can think of this like a fluid. And so therefore you can write down a fluid stress, which involves only the gradients of the velocity field. You can show that the pressure can be forgotten because there is turnover. And even if you include pressure, that just renormalizes the bulk viscosity. So I have equations for the density field, I have a flow field. So one should be able to write down consistent system of equations. Okay, so this is a busy slide. Too many equations, but then I'll ditch most of them. Just hang on for a few minutes. So you start with force balance, and wherein you say that I want to get the stresses. The stresses have contributions from the orientational degrees of freedom, like a polarity order parameter, have contributions from the fluid degrees of freedom, the standard Newtonian equations, and you write down a linear viscoelastic fluid, like a Maxwell viscoelastic fluid. Now, if you have an order parameter, like a polarity order parameter, you need to write down an equation of motion for the order parameter that involves certain coupling to the fluid degrees of freedom as well. So this is standard liquid crystal elasticity that we know from the 70s, 80s, and 90s. I can't hear you. Eta B is the bulk viscosity. I'm not assuming incompressible fluids, not standard fluid dynamics. On top of that, if you have a bunch of chemical species that are sitting there, things that are embedded in the system, obviously they'll be advected by the flow, can diffuse and have some chemical reactions coming up there. But the most important thing that makes the system non-equilibrium is the fact that these are things which have active stresses. The active stresses come because, like I said, the ATP hydrolysis drives the system out of equilibrium. And to a coarse-grained description, to the level of description that we are looking at, you can construct phenomenological stresses, which could have an isotropic part and an anisotropic part that comes from the polarity order parameter. So remember, there is no such thing called an isotropic active stress in 3D, that's just pressure, Pascal's law. But because I'm looking at a thin film, so I can have isotropic active stress in the 2D plane of the thin film. So the way the active stress enters is that will couple to the constitutive relation. But not just that, if you have a polar order parameter, there are a bunch of terms which are not allowed if you're at equilibrium. But if you break equilibrium and are active, certain new terms are permitted. But anyway, don't worry about that. This is obviously quite complicated, right? So usually one writes all of these things and then there's so many parameters, it's very difficult to understand what's going on. So what I'm going to do is I'm going to assume that there is isotropy in the cortical plane, so which means I'll neglect all the anisotropic components. I'll assume there's fast viscoelastic relaxation, so which means I'll neglect all the viscoelastic effects. And so what that does is to knock off a bunch of things, which is nice. And then I get something rather simple. So I have a fluid for which I write a force balance. The stresses come from hydrodynamics and activity. Hydrodynamics is just the viscous stresses. Activity, I assume, is isotropic active stresses regulated by the concentration of, let's say, myosin motors. That generates the flow. And once I have the flow, to solve the flow, I make a frictional approximation. I say the external force is just the force the cortex feels with the membrane. Once I have the flow, I can use the flow to transport material. But then I can take this constitutive relation, put it into the force balance, and so that get two equations which seem to be reasonably easy to solve. So I have transport plus reactions and flow, which is regulated by myosin concentration fields. So this itself, I want to demonstrate, leads to a rich set of patterns that can explain a significant number of biological problems. Okay, so most of the time I'll just look at even one species here, and for one of them we can even try to do a linear stability analysis. Can I go ahead? Yeah.
How does your free energy form look like? Is that a single strain energy form, or does it include the species as well?
Which form?
You write a potential to solve for this, right? So
or what?
Usually you have elastic potential, and
there is no elasticity. It's a complete fluid.
Okay.
Right. So I said I'm going to look at the limit where I have fast viscoelastic relaxation. So therefore, I'm beyond the elastic time scales. It's really flowing like a fluid.
It related to that. So the way to think about this is, can you help think about the fact that the underlying substrate is actually not undergoing any motion. So you're looking at this thin layer on top of
beneath the cell membrane.
Yeah,
it
has an underlying geometry on which this fluid lives. But as is the geometry of the structure itself doesn't change.
You mean the thickness changes?
The shape of the cell itself does not change.
For now, I'm assuming the shape of the cell is not changing. If you wait for an hour, I'll show you some shape change. Okay, so I have something where there is an equation for the flow. Think about this like the Stokes equation for the flow, where there is a source term, which is essentially the gradient of the concentration field of the chemical species. The chemical species moves because there is flow, diffusion, and maybe chemical reactions. So I want to show you that there are very nice rich patterns coming here. So already you can see something non-trivial will come up. So like I said, there are myosin motors which tug on actin filaments and generate contractile forces. So here's an intuitive picture. Say I have a patch of the cortex and there's some density of myosin motor protein. So on the average, this will lead to a contractile force, which means it will contract if you wait over time. So let's say I have another patch with a higher density of motor proteins. Surely the contractile stresses there are larger. Okay? So here's the conceptual idea. If this was actually a connected patch, in other words, I have a gradient in the density of the myosin motor proteins, surely that lead to a flow. Right? So therefore, myosin contractility gradients lead to flow. Does this work in the real cell? Okay, here's the proof. So this is a C. elegans zygote, very first cell division. What's labeled is myosin motors, the very ones that generate active stresses. So you're looking at the bottom plane, confocal imaging. So you see these patches of myosin flowing at the bottom part of the cell. And you see, if you wait for a few seconds, you'll see there's a persistent flow from the right to the left of the spots. So once you have that, you can do some imaging techniques and get particle imaging velocimetry and get the velocity field out. So what I have is a field of the source and I have a field of the velocity. I would like to check if my active mechanics can make quantitative predictions. So in one dimension, that is, I assume motion only along this direction and I average it out the y direction. So I can write force balance in one direction and get a flow equation where I say that the active stress gradients drive flows. So experimentally, I can measure the myosin profile from the real worm. And dotted points show the experimental flow profile. And the solid line and the dashed line show the fits of the theory to the experimental profile. So if you notice, the flow is in real units, microns per minute. The concentration is, of course, arbitrary, but then you get these coefficients like the material properties of the cortex, right? So this just shows that the idea of active hydrodynamics works in real living systems, the ones that are alive. But this also leads to a new class of patterns, and that's what I want to focus on. These are patterns that involve mechanics and chemistry in a very non-trivial fashion. For me, chemistry is the fact that motors generate active stresses. That's the mechanochemistry that I'm talking about. Okay, so here's the simplest model, and I want to illustrate this, if possible, with a calculation. I'll consider one concentration.
So, Vijay, one quick question. So, from the previous image that you showed,
Yes.
it feels like there is some kind of a spot-like formation, and
Yes.
and there must be some collapse in the concentration,
Correct.
strength, and stuff.
So,
that,
there's another story behind why there are spots. This one just neglects the spots and average it along the y direction and just look at the profile.
Okay.
You're right. So there are spots.
Good
It's not a
enough.
homogeneous thing. It's not a uniform gradient. There's another instability sitting at the back of it. I'll show that in a minute. I look at one concentration field. It advects, it diffuses. The concentration field C generates an active stress. The gradients of the active stress generate a flow. So the flow will feed back into the concentration field. Now we have a coupled system of equations between the concentration field and the flow. So for a second, if you look at this and ask yourself, suppose there was no activity, that is to say, there were no active stresses generated by the concentration field C, you can see in a second that the flow will eventually be zero. There's nothing else, everything is damped out. So therefore the advection term will be non-existent. And the only thing that changes C is diffusion, which is purely stabilizing. So therefore, if there are any non-trivial patterns that will come up, it must be because of the active mechanics and chemistry coupling. You can define a Peclet number, which is basically some activity to diffusion measure. And you can show, like we did yesterday, that the state C equal to C naught, in this case, C naught is some average concentration, and V equal to 0, that becomes unstable when the Peclet number becomes much larger than 1. So there's actually a type 2 instability. That means you have a growth rate versus the wave vector, and the largest growing mode is essentially the smallest wave number that's there in the system. So if you do a numerical simulation, you'll actually get a big clump coming and sitting there. Okay, so this is a spontaneous instability of the homogeneous pattern for large Peclet numbers. So like I did yesterday, if you include a simple regulatory chemistry, a linear degradation reaction, so you have one more length scale now, square root of d by k is another length scale, the same L1 that I showed you in the Turing patterns yesterday. This time it becomes a type 1 instability with a non-zero wave number where the instability happens, and you get a very nice hexagonal lattice pattern coming up. So these are the kinds of Myosin patterns that were there. So there's a large scale flow, but there are these patterns. Okay, so I want to emphasize, if you don't have the coupling to mechanics, there are no patterns in the system. This is like a minimalistic constructed system where the patterns come essentially because there's a coupling to mechanics. And you should really think about these as the mechanochemical analogs of Turing patterns, right? So these are patterns that come up in chemical concentration fields, not because there's nonlinearity in the chemical interactions, like in Turing, but because there's a coupling to active mechanics. Something that will turn out to be important towards the end of the talk is that there are patterns in the concentration and the flow. The flow is the arrows here. But not just that, there are patterns in the stress. And so if there are patterns in stress, surely I can use that to deform something.
What's the gamma in the second equation?
Yeah, this one. So I said that the cortex is some thin material beneath the plasma membrane. I don't know where I put that. And the external force from the cytoplasm and the membrane, I approximated to lowest order as a frictional coach, minus gamma.
Yeah. So that's basically the fluid equation, right?
Which one?
Stokes, similar to Stokes equation, right?
Stokes with a drag.
Yeah, but where is the pressure in that?
Okay.
Gradient
if
of
you keep
pressure.
making me go back, I won't be able to cover many points. So where did I have that? I did have it somewhere. Yeah. So I told you that if you include pressure, the fact that this is a system which is turning over continuously, you can show that including the pressure, and in the steady state, the including the pressure will just change the value of eta b and nothing else. Therefore, I can neglect the pressure. Remember, this is not an incompressible fluid. It's a compressible fluid. Yeah. No, there I had actually. These ones? Yeah. That's correct. Yes. Yes. Exactly. That's why I said this is a thin film. So you're looking patterns on the thin film. So wherever there's div v non-zero, material is going inward. That's why div v is not 0. The term that accounts for what? So the fact that there is a div v not 0, C is not the density of the material, C is the concentration of some other chemical things buried there. I've assumed there's a constant density, there's a continuous turnover that fixes the density, but the velocity field is non-zero, so stuff can go in the third dimension and come back. Div v is not 0 therefore. Okay, so what I want to do is illustrate to you a bunch of problems which takes such an approach and shows you, how they're sensitive to the boundary geometry, how they're sensitive to the curved surfaces and even dynamical surfaces. So give me a feeling. Should I work out the linear stability of this on the board, or can I just show slides and go ahead? Slides. I'm too tired for the board. Okay, fine. Easy for me too. Turns out that if you take this system and if you work out the linear stability analysis, at the linear stability analysis, you can show that even including turnover, the only thing you'll get is a stationary pattern forming instability. So that means eventually the patterns will reach some stationary state. The growth rate is such that the imaginary part of the growth rate is 0. There's only a real part of the growth rate. So, but many of these patterns are actually oscillatory. So we wondered at some time back, is there a way to get oscillatory patterns in such a system? So at that time, we thought that you cannot get it with one species, which seems reasonable, because at the linear stability level, you need complex eigenvalues, and you cannot get complex eigenvalues if there's only one species there. So you need a matrix to at least get complex eigenvalues. So it turns out that if you have two species, let's say an activator of the stress and an inhibitor of the stress. These are not the activator inhibitor of the chemical reactions like in Turing, but activator inhibitor of the active stresses there. So if you write down the flow equations, we could show by doing simple linear stability and explicit numerics that there is something like a Turing pattern, but for mechanochemical, Turing-like criteria, but for mechanochemical patterns. So if you don't have any chemical reactions, both just advection diffusion, you get an oscillatory instability when the active stress upregulator, the activator, diffuses rapidly compared to the active stress downregulator. It smells like Turing, but it's not Turing. The structure is very different. There's mechanics sitting here. Or you include turnover, and let's say keep the diffusion constant the same, you get oscillatory instability if you have the activator turning over faster, kappa A is greater than kappa I, and the downregulator turning over slower. This is something like a Turing-like criteria, but for pulsatory pattern that automatically gives you pulsatory patterns. Okay. So like I said, if you have one species, you would normally think that oscillatory patterns, waves are not possible. And that's mostly true. But something that we discovered some few years ago, and it took us some time to understand why and to demonstrate it analytically. And I want to show you this. So this just appeared a couple of months ago, early part of this year. So let's say I have only one species. And I have my advection, diffusion, turnover, and there is the fluid velocity. So I've just non-dimensionalized things. So the only three relevant parameters are the Damkohler number, which is essentially the reaction rate, the Peclet number, which is essentially the strength of activity, and something that measures the viscosity. Okay. So if you do a linear stability analysis of this around the C equal to 1 and B equal to 0 state, easy to show that the linear stability growth rate, what I called omega, I'm calling it lambda here, has this form. And now clearly there's no way that this can become complex. So therefore there are no possibility of oscillations. In fact, if you look at the growth rate is always negative at 0 wave number and can peak up and go down as you increase the Peclet number. And there's the linear stability phase diagram. You have some homogeneous boring state here, and you have a pattern state, like the pattern state that I showed you earlier. Okay. If you go further and do what's called an amplitude equation, I did not cover that. I hope people have seen that in some fashion, maybe Ginsburg-Landau, something like that. There is a way to look at the analysis of patterns, not just at the linear stability onset, but slightly beyond what's called amplitude equations. You can do that for this and show that you essentially get the real Ginsburg-Landau equation. And so therefore, that also predicts that you will only get stationary patterns. Both linear stability analysis and amplitude equations show that the homogeneous state will undergo a stationary instability. So therefore, no oscillatory patterns, right? So we did numerics, And to our surprise, we actually found traveling wave patterns in the system. So for some time, I mean, I thought there's some bug in the code and this is not fine. And you change the method, the numerics, the mesh, the student, you do all those changes, but the pattern survives. So therefore, then maybe there's something real there, right? So it seems to be that it is independent whether you have periodic boundary conditions or no flux boundary conditions. If you have periodic boundary conditions, of course, you get traveling waves. But the analog of that in the no flux boundary would be sloshing waves. Okay? So that seems to be persistent there. One wonders why and so where. So if you look at the average dynamics, let's say you integrate the concentration over the domain. So I'm showing this in 1D for simplicity. I'll show you something in 2D in a minute. If you integrate the concentration over the domain and write the corresponding equation for the average, you can show, it's a trivial, boring exponential decay. So therefore, any time-dependent pattern that must be coming should really be because of the nonlinear coupling between spatially inhomogeneous modes. It cannot be something which involves the homogeneous mode. So how do you capture that? Now, the linear stability already told us that you cannot get oscillatory patterns. So therefore, this must not be something that happens at the linear stability level. This is probably some nonlinear instability that's happening. But how do we understand that? So in Fourier domain, in periodic domains, you can do a Fourier expansion. So write the concentration in some Fourier expansions with modes, amplitudes, and phases there. And I just remarked that there are no bulk oscillations possible. So therefore, for n not equal to 0, is there a way I can develop a theory to show that there is indeed some untrivial oscillatory patterns coming up? Okay. So there's a bit of algebra. So if you take this system and let's say you truncate this, that is, I do a Galerkin truncation, I assume that there are only two modes which are important. I take that expansion with involving two modes, I plug them back in, do some bit of algebra, do all the integrals, blah, blah, blah. And at the end of the day, I can show you that there are equations for the amplitudes, ρ1 and ρ2, which are like that, and the phase difference 2 phi 1 minus 2 phi 2, which is like that. So what we did by doing this is went from a partial differential equation system for C and B to a system of coupled ordinary differential equations for the amplitudes ρn and the phase phi. So there are a bunch of parameters, alpha, mj. There are basically something which are geometry and boundary condition dependent terms. There are no dynamical variables sitting there. But the more important thing is the following. So we got these equations, but this is a system of ODEs. Now, once you have a system of ODEs, like we discussed yesterday, you can find the fixed points, you can find the stability of the fixed points, you can do bifurcation theory, and so therefore you can get the phase diagram of the ODE system. So we predicted that if you look at the homogeneous state and ask where is the homogeneous state existing and where is it stable, that is, you look at the homogeneous state, do linear stability, find out the eigenvalues, positive, negative. You find the homogeneous state exists here and is stable. In this region, it's unstable. The stationary patterns don't even exist there. They exist here and here, but they're stable here, but unstable there. To our surprise, we found that there are traveling wave states. The traveling wave states are characterized by the fact that the amplitudes are non-zero and the phase lies between 0 and pi. So that state exists and is stable. So if you combine all these three, here's our prediction from the analytical Galerkin model. Say there's a homogeneous state here, there's a stationary state there, and there are traveling wave patterns. This is for 1D periodic domains. If you do numerics, you get something very good. So even the numerical value of the parameters for the Peclet and the turnover are quite close by. Look at this. So here's some snapshots of the patterns in the stationary regime there. You have in the traveling regime. In the numerics, if you go to very high Peclet, in addition to simple traveling waves, you also get oscillatory traveling waves, much more complicated things. So surely there are more bifurcations going on as you go up in Peclet.
Quick question. So the homogeneous to stationary state, that line?
Yes, that's linear stability.
That's the linear stability of the constant.
The homogeneous state.
Why is that not matching with the numerics?
It is matching with it.
It is crossing at 2 over there and crossing at 1 near 1 over here. I guess the y-axis is different. Okay.
Y-axis are different.
Okay,
right?
so those lines are identical, but everything else is.
Linear stability, everything works. Linear stability is a homogeneous state.
Why do you not capture the oscillations with the Galerkin model?
So remember, this is a two-mode truncation, right? So clearly, if I have something more than a simple traveling wave, I need many more modes.
Yeah, so it's just two mode.
It's just two mode.
Okay, yeah.
So, and there's one thing which I want to illustrate. From the numerics, we find that you can never get traveling waves at 0 turnover. And in the paper, we have an argument to show why it's not possible at all. But from the two-mode Galerkin truncation, it already predicts that there is a possibility of getting. So this is an artifact of the truncation. You've done one check that, let's say, you include one more mode, 3 mode. This intersection point goes up. So I suspect that the more modes you include, the higher it goes up. But then, the more modes you include, the more algebra, it becomes horribly complicated.
One quick question regarding the speed selection for the traveling wave.
I'll show you some plots.
Okay.
Okay, so first thing you notice is that the concentration profile breaks the fore-aft symmetry. There's a traveling wave. So if you look at the peak of the profile, it's not reflection symmetric about the leading and the trailing edges there, right? And if you have a non-zero phase difference between the two modes, well, clearly the profile that you would construct like so will not have centrosymmetry. There's a phase difference between 0 and pi. That's something we see in the numerics, and that's something the Galerkin model also predicts very well. Sorry, Ganga, I may not have the phase plots. I'll see, I don't remember. So if you take the domain, make it, let's say, double the size, well, you get 2 peaks. So this is not something like an advective instability. This is actually something where there's an intrinsic length scale. Like I showed you, if you have no flux boundaries, you get sloshing waves. Seems to work in 2 dimensions with periodic boundary conditions, also with no flux boundaries that we have seen in the numerics. This is something that depends on the flat geometry. I mean, you'd like to understand that. So we wrote the same model on the sphere, on a surface of a sphere. So on the surface of a sphere, you have the concentration equation, which is very similar, but the flow equation is a little more complicated. First of all the grads here are the covariant derivatives. I assume people have done covariant derivatives and Christoffel connections and so on. It turns out that if you write down the flow equation, you will get an extra term from the Gaussian curvature. Okay, and this is essentially because the covariant derivatives don't commute, and the commutator of the covariant derivatives is basically the Riemann curvature tensor, which for 2D, the only invariant is the Gaussian curvature, and that appears naturally. Now, to analyze this on the sphere, you can choose the appropriate spherical expansion, spherical harmonics, where you expand the concentration field. But remember, I also need to solve the velocity field. So just like you have scalar harmonics, you have vector harmonics. So there are 2 vector harmonics sitting on the surface of the sphere, reasonably well known. One can work out all the equations and the relations. I'll skip a bunch of algebra, and once we do all that, we can show that if you retain still the 2-mode truncation, we get the same equations as we got in the case of 1D, which is remarkable. The structure of the equations is the same. Some algebraic coefficients here and there are different depending on the surface of the sphere. So it looks like, the kind of instability that we've discovered is something which is independent of even the geometry. So here's the analytically predicted phase diagram for the sphere. Here's the numerical phase diagram for the sphere. As expected, if you go to higher Peclet, there are more bifurcations, there are oscillatory patterns and more complicated things that are coming up. So here are snapshots. Better, I show you movies. Sorry. So there are traveling waves starting from a homogeneous random initial condition. The direction is randomly picked up. But there are more complicated patterns. There seem to be oscillatory patterns if you go to much higher Peclet. There are 2 spots, and the spot seems to go back and forth over time. There's some ongoing work where we have seen how the geometry of the surface matters. Of course, we can't do analytical calculations, but we have a very nice way to do numerics on any arbitrary surface. And I'll discuss the numerics as we go along. So we are trying to explore how such patterns are sensitive, mechanochemical patterns are sensitive to the geometry of the underlying domain. Do the patterns select regions of high curvature, low curvature, go to regions of negative curvature? We would like to explore that systematically. Okay. Turns out that this is this instability that I discussed, the secondary instability, is related to non-reciprocal interactions. One thing to remember is that the waves that I showed you do not come from a Hopf bifurcation of the homogeneous state. Like I showed you, the homogeneous state just undergoes a stationary instability. It's really the nonlinear interaction between the inhomogeneous modes that generates this intermode phase difference. And one way to understand such kind of patterns is to ask, if you have one-dimensional cellular patterns and you have a system where you break the parity of the patterns, let's say you put some antisymmetric perturbation, it was known quite early that if there is a parity breaking perturbation, you can lead to a net translation of the whole pattern. And if you look at the equations of motion, we can exactly identify the non-reciprocal interactions between the two modes. And if you switch off these, of course, you don't get traveling. These are the ones that are actually causing the appearance of traveling. So there's a clean analytical demonstration that non-reciprocity leads to waves independent of 1D, 2D, or on the sphere. And it's really the non-reciprocal interactions that come from the activity that matter. All right. This is nice. This is all theoretical. But does it work in real systems? I showed you one example. I want to show you one more example where such active mechanochemical patterns are useful in real biological systems. This involves a little bit of biology. I hope you'll bear with me. This I showed you yesterday, that there are patterns that are possible during cell division where the two daughter cells are different. The difference could be size or composition, asymmetry, and so on. Turns out this asymmetric pattern is driven by certain patterns that appear on the surface of the cell. So like I told you, these are the ones that come up earlier. The output of this is where the cell decides where to divide. This involves both fate determination and axis establishment. This is the pattern I want to show you is actually an active mechanochemical pattern of the kind that I've been discussing. So this is a pattern where there are large-scale redistribution of certain proteins. Remember, this is like 50 microns in size, driven by large-scale flows that appear on the surface of the cell. So these are the same flows for which I showed you active mechanics works quantitatively, but these are things that are happening on the surface of the cell. How do we understand them? If you want a quantitative understanding, surely you need quantitative data. So this is something that we did as a collaboration with Peter Gross. We can take microscopy images like that, look at the cross-section, identify the peak of the concentration, so therefore digitally cut it open and get the profile along the periphery of the membrane. As far as we can make out, the pattern seems to have azimuthal symmetry, so therefore you can just look at one mid-plane cross-section, and that seems to be enough. You can calibrate it against known intensities and so get actual number of proteins per micrometer square on the surface of the cell, of the red proteins and the blue proteins. You can do the same thing for myosin, which is the motor protein causing flows, but you can also do particle imaging velocimetry. So at the end of the pipeline, what we have is a way to convert microscopy images like that into data like that. So these are space-time fields evolving. So you have a red field, you have a blue field, you have the myosin, and you have the flow. You see the red starts out being more or less homogeneous. At some point, a blue domain develops, a red domain clears out, and the accompanying flows establish the polarity pattern. We write down a theory for this involving the two proteins that make up the red and the blue pattern-forming systems. You see the same structure, advection caused by active flows, diffusion, and some regulatory chemistry. The regulatory chemistry will, of course, depend on the particular molecules that you are looking at. So that's some non-generic structure there. The active mechanics module involves the myosin motors, advection, diffusion, chemistry, the same velocity equation that I showed you earlier, where contractility is generated by myosin, gradients of contractility drive flows. The flows are the ones that separate this. I won't go through the details. Surely, as physicists and mathematicians, we should all be puzzled. Like there are so many parameters, you can fit an elephant. If you have 5 parameters, that's lower in physics. We are well aware of that. So I've skipped like 10 slides before this, showing you that there is a power of the genetics of the worm, where we can selectively knock out mechanics, selectively knock out polarity, and then fix like 3-4 parameters, and then therefore fix all the 11-12 parameters individually. We can solve the system. We can compare the data, we can compare the numerical solution with the data, and the matching is so good that we can actually predict the spatio-temporal evolution of the pattern on the surface of the living cell, okay, including actual number of molecules per micrometer square on the surface of the cell. So this is just to show you that this structure and this class of patterns, something that works very quantitatively in real systems. These patterns are very sensitive to geometry. Is where the geometry aspect comes in. And I want to understand that. I'll start out with something simple and then let's go to more complicated geometry. So remember the system of equations that I showed you? Let's say I take this system and put it in a 2-dimensional domain, flat 2-dimensional domain for now. And I impose no flux boundaries on the edge of the domain, where I demand the normal component of the gradient of C as well as the velocity vanishes of the boundary of the domain. So I'd like to understand the kind of patterns that will come up. and if the patterns are actually sensitive to the shape of the boundary. So this is not unrealistic. You can actually take cells, you can plate them on substrates, and you can engineer micropillars on substrates, and you can confine cells in whatever shape you want. It's pretty easy to do with 3D printing these days. One of the things that we were able to show is that if you take this system, do a Galerkin analysis just like I showed you earlier. Remember, this is a system with boundaries, so you don't have translation invariance. So the modes that you will choose for doing Galerkin analysis will have to be the eigenfunctions of the Laplacian on a bounded domain. In this case, some Fourier-Bessel things. It's boring algebra, but can be done. And we predict analytically that there will be a homogeneous state, there will be an anisotropic state, something like this, where there's a peak concentration at one of the edges, and something where there is an isotropic state. Numerics also shows something very, very similar. If you perturb the geometry, let's say rather than a circle, you make it a mode 2 deformation. I can't do anything analytical. I don't know eigenfunctions of the Laplace for arbitrary geometries, but I can do numerics. And what we see is that there are patterns that seem to sense the boundary curvature. There are parameter regimes where the patterns go to high curvature, low curvature, or they're more or less uniform on the surface, and so on. The thing that I want to emphasize is that these patterns sense geometry. And this is something people have observed with actin patterns. And depending on the parameters, you can actually get patterns that localize to higher curvature regions, lower curvature regions, and so on. You want to think about geometry where there's some non-trivial curvature as well in the game. I take the same model, that is a concentration field C defined on the surface of the cell. Remember, the red is the cortex of the cell. I have advection, diffusion, simple linear turnover. I write down force balance on the surface of the cell. There are the passive stresses, there are active stresses. And I say I'm going to include the cytoplasm as well. So for simplicity, I'll think of the cytoplasm as a simple, boring, viscous fluid, incompressible fluid for now, the 3D incompressibility. And so the way I can write down the fluid dynamics of the 3D incompressible fluid is that I wrote down Stokes. Here I include the pressure. This is the pressure in the bulk of the cytoplasm, incompressible. The boundary condition is that the cytoplasmic velocity at the surface is equal to the cortex velocity that you have obtained from the active mechanics. Secondly, the stresses that you generate in the bulk, the stresses, the normal component of the stresses evaluated on the surface give you the traction force. So if you remember, I had put gamma V earlier, that was an approximation. This is the full story where you can actually evaluate the total stress, look at the traction force on the surface. So now you need to do vector harmonics on the surface and vector harmonics in the bulk as well. Again, a bunch of algebra. Once you do that, something that we predict turns out to be nice. So there's a length scale that comes up very naturally, which is the ratio of the shear viscosity of the cortex, the surface, and the shear viscosity of the cytoplasm, the bulk. So you can see that these two are not the same units, and the ratio is actually a number. So the comparison of this length scale with the radius of the sphere is what dictates the kind of patterns that you will see. If you have this length scale being much, much greater than R0, turns out you get a L equal to 1 instability. Remember, this is the L of the spherical harmonic cell. So if L equal to 1, you get an instability where there is a single spot. If this length scale is much smaller compared to R0, you can show that the first instability you get is at L equal to 2. So pictorially, this means that you can either get a cell polarity-like pattern or, surprise, surprise, a cell division-like pattern coming out very spontaneously on the surface of the cell. Okay. However, for the realistic C. elegans zygote, it turns out that this ratio is of the order of 10, so we're actually in the wrong regime. Now, you would like to probe the nonlinear regime. Remember, these are all linear stability predictions. And so you would like to find out if you can do robust numerics for any arbitrary geometry. That's what we've been doing so far. Okay, so I'll take a minute to explain the approach that we take to solve all these partial differential equations on arbitrary geometries. I'm gathering that you've done differential geometry. You've seen things like covariant derivatives, Riemann-Christoffel tensors, Riemann-Christoffel connections. So therefore, I'll show a slightly different approach. And if you have questions, that'd be very nice to understand. So I want to do calculus of fields on arbitrary surfaces. And the way I want to do it is by thinking about this as being embedded in an external space. Okay. So if you have an external space in which you have the surface embedded, I'll assume the surface is oriented, so which means there's a well-defined normal at every point. So if I have a normal, I can construct the tangential projector. And the tangential projector is a way to get the tangent plane to the surface at every point. Does that make sense? The way I want to think about fields living on the surface is that I will think about the fields as having a small extension in the ambient space. I will do Cartesian derivatives, hit it with the appropriate number of tangential projectors, and show you that you get the correct covariant derivative. So here's an example in one dimension. Let's say you want to solve some PDE on a curve, and there's some concentration field there. The color is supposed to represent some concentration field. The way I do that is by saying I'll extend the field into the ambient two-dimensional space. Of course, there are no unique way of extending that. Turns out it doesn't matter so much. Here's one easy way to extend. You do a constant normal extension. So you take a field, maybe scalar, vector, whatever it be. You don't vary the field along the normal to the surface, and you extend it by an epsilon amount above and below the surface. So that gives you a genuinely two-dimensional field in this case. So now I can do two-dimensional Cartesian derivatives, and then I'm going to hit with tangential projectors. For a vector field, if you have a vector field here, so the way you would do the extension is, extend the field in a constant normal fashion above and below the surface, genuine 3D field, construct your derivatives, and hit with projectors. So if you have a concentration field, which is a scalar field, from points on the surface to real numbers, the surface derivative you can show is if you take the 3D Cartesian derivative and hit it with the projector once. This gives you the correct surface derivative of the concentration field. Scalar fields are easy, but all hell breaks loose when you go to vector fields, as you would have learned in differential geometry. So if you have a vector field, it's easy to show that you take the 3D Cartesian derivative of the vector field and hit it with projectors on both sides, and that gives you the correct covariant derivative of the vector. Okay. So therefore, what we have is you give me an arbitrary surface, you tell me the normal direction to the surface at every point. If I know the form of the surface, I can calculate the normal if analytically or even numerically. Once I have that, I have the projector, and so therefore I have constructed the calculus of scalar and vector fields sitting there. So this is a very robust method, and it's easy to implement this on a finite element network. I gather you also learned finite element exterior, or at least discrete exterior calculus. So something like that goes on here. So you can look at this approach on any arbitrary meshes, even of different topology. And if I have time, I'll show you even dynamical shapes. In addition to the derivatives, you can, of the scalar and vector fields, you can look at the derivative of the normal vector itself. And as you learned, this is basically the curvature tensor. This is in fact the extrinsic curvature tensor, shape operator. So the eigenvalues of the curvature tensor, it has one null direction, which is normal. The other two eigenvalues are the principal curvatures, and so therefore you can define the mean curvature and the normal curvature. So if you give me a surface, you can specify the surface completely by giving the normal and the curvature tensor everywhere on the surface. So I can reconstruct the curvature tensor exactly. Okay, so all calculations are done in the embedding R3 space, so there are no coordinate singularities that you have to worry about. If you don't know this, it's okay. If you know this, you know what hell of a problem are coordinate singularities. Now, what's the use? I'll take my model, I'll write this on arbitrary surfaces, and now I'll ask, how does the geometry of the surface matter for the patterns? So simplest deviation from a sphere, I take spheroids. So this is an oblate spheroid, so the axis of symmetry is the vertical axis, so it's azimuthally symmetric about this pattern. So if I take this model and run the simulations, I find that depending on the Peclet number, basically the activity, the localization of the spots seems to be correlated with the curvature of the surface. So the spots seem to go at high curvatures for low activity, and they seem to go to low curvatures at high activity. Same thing if I do a prolate ellipsoid, they seem to go to low curvatures at high activity. Remember, the high curvature is the pole now, and the low curvature is the equator coming up there. So this seems to be a nice phase diagram, and there's a nice smooth transition that happens there. So we are trying to understand this more quantitatively and trying to do some semi-analytical calculations. All right. Last part, and this is the most recent piece of work we just put out on bioRxiv about 2-3 weeks ago, something that we are very excited about, and I want to show you this. How many of you have heard the word geometrodynamics? You heard? In what context? Louder.
I may be wrong. I've heard it in the context of geometric elasticity, but I guess it has something to do with general relativity. Yeah.
Okay. Thanks. So this is a collaboration with Masatoshi Nishikawa, who did the experiments, and my PhD student Aditya, with whom we built this story. So I'll take the next 20 minutes to explain this, and I want to go slow, and I want to show you some very nice results. Okay, we go back to cell division, and this is a picture that I've shown you. The cell division not only accomplishes the segregation of the genetic material, but it's also a problem where there are dynamical shape changes going on, right? So that's the mother cell, which is more or less spherical to begin with. It develops a constriction, a peanut-like shape, and then an eventual cleavage into two daughter cells. So like I said earlier, this is accomplished by a large-scale actomyosin ring that forms on the surface of the cell, and the constriction of the ring is what actually leads to the cleavage of the cell. So here's a picture that we know. Somehow flows seem to generate an actomyosin pattern, like a ring, and the ring is a ring of actin and myosin, so therefore there are contractile forces, and the contractile forces lead to the constriction. That's the classic picture of cell division that we have here. I want to tell you two things. One is that the asymmetric cell division that I talked to you earlier, where the two daughter cells are different, which is driven by certain patterns on the surface of the cell. Okay. But at the same time, there's another asymmetry, and that I want to bring to your attention. So look at the same movie again. You see the asymmetry in the two daughter cells. Fine, I'll talk to you about that. But there's something funny that happens. If you actually look at the membrane of the cell, the surface of the cell, you see something weird. One side ingresses more compared to the other side. Can you see that? Okay. So this is not standard classic textbook cell division where there's a nice ring and the ring constricts. If you look at the division plane, you can really see the cytokinetic ring ingressing, but also moving in an asymmetric fashion. So this is the time course of the evolution of the ring. So this is in the C. elegans embryo. Maybe this is something specific to the worm. Living things are weird. They have things for no reason. Turns out, no. This seems to be something that is seen across many species of cells. Here's an old picture from Rappaport's book where he shows some qualitative ideas of how the sand dollar does this ingression. This is a video I lifted off Twitter when it was called Twitter. You see, this is calm jelly. The ingression happens only from one side. The other side does not ingress at all. Something you see similar in the C-star embryos. Here, the spindle is localized to one side of the cell. So there seems to be some generic thing about why asymmetric ingression happens, and we would like to understand the physics of that part. In C. elegans, as far as we can make out, this is something many others have done. We have also reconfirmed this. The orientation of the asymmetry, that is which side it ingresses asymmetrically, seems to be random. You repeat the experiment many times, there doesn't seem to be any signaling which is dictating the direction. It seems to be persistent even if you put the embryo along gravity. Maybe it's because you squish the embryo like that and that's all. No, you put it along gravity and the cross sections are much nicer and circular, but the ingression is still asymmetric. As far as we can make out, the spindle is more or less at the center of the cell throughout the ingression process. So to a first degree, we can probably neglect the spindle for the ingression. We've reconfirmed all this with our collaborator, and this seems to hold. What our collaborator did was to get myosin images. Can I switch off the lights a little bit, if possible? This is a dark background movie, at least the front. What my collaborator did was to image myosin by labeling it with some GFP tag myosin, and then get 3D confocal stacks of myosin in the cell and follow it in time as the cell divides. It's a fantastic movie that not many people can get. So you see three-dimensional constriction of the cell. You see a myosin pattern on the surface of the cell. A ring-like pattern emerges. The ring constricts in 3D. You also see asymmetric construction of the ring. Asymmetry seems to be towards the top side for this case. So we have plenty of movies like this. Once you have movies like that, well, digitally you can turn them around here and there, and you can go to the division plane, you can reconstruct the data, and so therefore you can get actual quantitative data of how myosin looks like on the ring, how the ingression rate looks like in the ring. You can get time course of all these patterns. So you have very quantitative data. if you have quantitative data, it begs a quantitative theory. You would like to understand why and how this comes up. So this is a conceptually different problem, and I just want to take a minute to explain that. So far, the kind of patterns that we've been discussing are patterns, maybe Turing patterns, maybe active mechanochemical patterns. These were patterns on fixed domains, right? Maybe periodic, maybe no flux, maybe sphere, maybe ellipsoid, whatever. The geometry was fixed, and these were patterns on fixed geometries. The cell division is a different problem. The cell division is a problem where the geometry is changing with time. Does it make sense? And the change in the geometry is not something externally imposed. It is something which is emerging in a self-consistent manner from the forces in the system. So there are patterns on the surface that changes geometry. The geometry changes the forces back and so on. If you make it such dark, people will go to sleep and I can't see who is awake. So not at least few lights, at least at the back or somewhere. Great, okay, thanks. So clearly there must be patterns on dynamical shapes, but not just that, there must be a crosstalk, right? So you would like to understand how patterns change shape, how shape affects patterns, and the two-way coupling the loop. So I've shown you one thing. I've shown you, if you have non-trivial shapes, how shapes affect patterns if the shapes are fixed. So I want to figure out how the patterns affect the shape and shape affects the pattern and so on. So this is what is called geometrodynamics. So there are fields on the domain which are giving you flows which come from stresses, and the flows change the domain, which changes the patterns, which changes the domain. So therefore, there's a consistent loop between shape and pattern. So there's a stress tensor and there's a geometry involved. Now, the traditional way of doing this is to put some intrinsic coordinates, and I assume you've all seen this: tangent vectors, metric tensor, normal, extrinsic curvature tensor. You do all that. And when it comes to covariant derivatives, oops, it's a bit complicated. So this is not my invention. This is well known in tensor analysis. This is very nice. But then if you want to look at evolving surfaces, surfaces that change with time, well, you typically prescribe some velocity field, and let's say you decompose the velocity into the normal and tangential components, and then you ask if the velocity is deforming the surface, how do things like the metric tensor and the curvature tensor change with time and space? And this can be worked out. I mean, this has been done in various versions since the time of Riemann and Gauss. I won't even quote. There's huge number of literature, and you can work that out. It's complicated calculus, but can be worked out. But then, you work out, you write the equations, and then you solve it for a sphere. That's not what I want, right? You would like to solve for some non-spherical shape. So much so that people have, seen that these are highly nonlinear, resort to numerics. And so therefore, if you go to numerics, there's no single parameterization for a spherical topology. I hope you know this. Theta equal to 0, theta equal to pi are a problem there. So you'll get stuck with coordinate singularities and so on. So much so that people have expressed their frustration even in papers. You write all these equations. The complexity of the equations may explain why they're often written but never solved. You write the equations but don't solve them. And so most of the times, you do some small variations around a simple geometry like a sphere, and you do perturbations, and numerical simulations are very, very hard. Okay, so what we did was to take some time, a couple of years, and develop a robust method to solve this problem numerically in a consistent manner. So just like the tangential calculus that I mentioned to you earlier, so think of a surface, and on the surface, I have my normal vector field defined everywhere. Assume simplicity orientable surface and a closed surface for now. So there's a unique outward normal well defined. So the variations in the normal on the surface defines my surface curvature, the extrinsic curvature tensor. So remember, all grads are covariant derivatives defined like so. The velocity field that will deform the surface, I'll take it to be something which is perpendicular to the surface and parallel to the surface. Now, when I describe the physics of the actomyosin cortex, I'm thinking about this like a cell, and at the periphery of the cell, there is the actomyosin cortex. When I describe the cortex, I told you I'm looking at timescales longer than the elastic relaxation timescale of the cortex. So therefore, I can assume it's a viscous fluid. So what I have is a thin film viscous fluid as the surface that I'm looking at. So how do I write the equation of motion? Well, obviously, it's only the normal component of the velocity which will move the points on the membrane. Does it make sense? There are no in-plane elastic stresses. So it's only the normal components that will move the membrane. Now, once the membrane changes, you can ask, how does the geometry change? So you can work out and show that the surface derivatives of the normal component of the velocity is the one that changes the normal unit vector. So the equivalent version of this, if you write down for the projector and the curvature tensor, will be essentially the dtGAB and the dtSAB that I showed you 2 slides ago. It's exactly the same thing, but written in an extrinsic description. Okay, so this is flow. This is geometry change given flow. So where do you get the flow from? Well, surely from the active forces on the surface. But before we go there, can we do something where you can check if you got it right and so on? So let's look at the deformation aspect. So if you had a passive membrane, you know the following, that in standard soft condensed matter elasticity, you learn something called the Kahnehammer free energy for a membrane, bending rigidity of a membrane. There's a way to formulate that in the language of a stress tensor, a bending stress tensor. And once we do that, we can go back to our picture and say that, look, I'm going to write down a stress tensor that has a hydrodynamic stress and a bending stress. And the divergence of the stress balances my external forces. That gives me a velocity. I'm going to use the velocity to move the points on the surface. And so that feeds back onto the membrane energy. So this is a problem where I can solve some things analytically. I know the eventual shapes of this. Let's say if I have volume conservation, is just a sphere. And so therefore we can recapitulate all that. So that means the way we are doing the geometry change is correct. So now all we need to do is to put in atomizing contractility. Clearly the way you do that is to put in an active stress here and make the active stress a function of the concentration field of myosin. So here's the full story now. So on the surface, we have a concentration field C, which moves by advection and diffusion and a simple boring linear turnover. The concentration field generates an active stress. So P is the tangential projector. That's the equivalent of the unit vector on the surface of the sphere, unit tensor on the surface of the sphere. The total stress will have something coming from the viscous stresses, grad V, bending stresses, like in the previous slide, and finally the active stresses coming from the concentration field. So I put them all in, do force and torque balance. I put one extra condition that I'll make the volume fixed, so which means I'll put a Lagrange multiplier, which will maintain a pressure difference between the inside and the outside. No area conservation, I'll just put in volume conservation. Couple that with the change in the geometry that I described earlier. Points move because there are normal component of the velocity, normal changes because there are surface gradients of the normal component of the velocity. So that's a coupling, right? So this system, I know if I have a static domain, gives me non-trivial patterns. This system, given velocity, it will change shape. I ask, can patterns and shape change dynamically and self-consistently? And in particular, what are the kinds of patterns that can come up? Yeah. That's right. We've done simple Euler, we've done predictor corrector method, IMAX methods. I can show you convergence plots and they seem to work reasonably well. There are a bunch of simple tests that we can do. If you switch off the activity, I know how it should go, I know the exponential rates. Those things seem to work out. Okay, so here's the conceptual picture that I have. So I want to start from something which is, let's say, a sphere and no patterns on the concentration. Patterns in both the concentration, velocity, and stress patterns will come up. The stress patterns will change the shape. Well, the shape will change the patterns. The shape will, so there'll be a feedback. So that's the thing that I want to ask. So I want to ask, what are the patterns of both myosin and shape simultaneously? So one of the things that we got, which is very interesting, is that we got cell division for free from this system. So we took a sphere, put some concentration, homogeneous concentration, small fluctuations. This is on a log scale to amplify the fluctuations. This is showing you the velocity field. As I run the movie, you will see vectors, which will tell you the tangential component of the velocity field. The heat map will show you the normal component of the velocity field. We found that there is a regime of parameters where the homogeneous state becomes spontaneously unstable, and you start getting ring-like patterns of concentration field and concomitant cytokinesis-like patterns of the shape. Okay, so the movie goes backward and forward to show you the thing a couple of times. You see the formation of a ring here, the ring ingresses, and the shape changes consistently. So here's a sequence of snapshots of the whole image. Hold on, 2 slides. I don't know if you noticed, the pattern is actually higher on one side, on the other side. 2 minutes. So we want to measure things. I mean, does it work in the real system? So let's say you look at this shape-changing thing, you define the transverse plane, the division plane, and would like to measure them. Remember, I said we have very good data from the experiment. We have chymographs of shape and pattern in space and time. We would like to compare them. So here's from the, So this is the division plane and the transverse plane. So as you go forward in time, you'll see myosin patterns accumulating there. You see more myosin here. Yes, you're right. This is still the symmetric ingression that's happening. So there's a whole phase diagram. So there's some non-dimensional contractility and there's some non-dimensional turnover rate. Clearly, if you're at very low contractility, the only pattern that's possible is a sphere with a homogeneous pattern of myosin. If you increase the contractility, you get a shape which is non-spherical with a single spot sitting there. And if you go to higher contractility, you start getting ring-like constriction patterns appearing spontaneously. This is an instability of the homogeneous state. But can we compare with the cell? Okay, so this is qualitative, this is nice, but does it work? Is the physics right? From the theory, we can, in the division plane, we can look at the chymographs of myosin. So this is time going down and you see myosin increases. This is from the experiment, not very impressive. Okay. Same with the ingression rate. Again, you do see the ingression rate coming up at 2 spots in the theory, a single spot in the experiment, but you do see an ingression pattern coming up in the experiment. Well, let's do some quantitative measures of this. One of the things that you can measure is the radius of the ring and follow the radius as a function of time. Our theory predicted that the radius goes down in some manner, but there is a regime where the radius goes down linearly with time. Okay? And the rate at which the radius goes down with time, U perp, increases with the Peclet, the contractility value. Can we test this in the experiments? Even before we went to the experiment, we noticed that several people had reported this feature across different species of cells. Starting from bacteria, sea urchins, and the worm itself, many people had shown that the radius of the diameter, there is a regime where the radius of the diameter goes down linearly with time. This is not something that we put in. We just put in active mechanics and dynamical geometry, and this feature just popped out. Okay? So the cytokinetic ring ingresses at a constant rate. And as I said, the ingression rate increases with activity. Can we test this in the experiment? So first we take the wild type, that is the naturally occurring creature, and then ask how does the radius go down with time. But C. elegans is cool because there's a lot of genetic perturbations which are known. People know that if you knock off this gene and that gene, the viscosity goes down, the velocity goes up. We don't understand why. We don't understand the map from genes to phenotypes. But there's a library, it's available. So there's a whole plethora of perturbations that one can do. And compared to the wild type, you can make the flows stronger or slower. So here's the wild type in red. So there's a prediction that the ingression rate will be constant and the radius will go down linearly with time. True. There's also a prediction that the rate of ingression should go up with Peclet. So GSP1 is a mutant which actually has a higher velocity. So this is cortical flow speed as a function of the genetic perturbation. And lo and behold, the slope of the straight line does go up in magnitude. Non-muscle myosin 2, an RNAi of non-muscle myosin 2, well, obviously will reduce the flows. Muscle myosin 2, NMY2 is the motor that generates the contractile flows. Clearly the slope goes down. Okay, so that's something that comes out from the experiments as well. Second thing, we can look at the myosin in the ring. Once you have the ring, you can ask how does the myosin accumulate in the ring? Remember, the ring is constricting, the radius is going down, so therefore the amount will go up. So in the theory, we found that there is a regime where the myosin increases exponentially with time. So that sort of defines the exponential rate constant Km. Surprise, surprise, even in the experiment, we do see a very nice exponential regime increase of the myosin. So the myosin accumulation rate, the Km, the prediction from both the theory and the observation from the experiment is that it increases with activity. Higher the activity, faster the accumulation of myosin in the ring. But remember, faster the accumulation, faster the ingression, because there's a nice feedback, there's a positive feedback that's coming up. So there's the rate at which the radius changes and there's the rate at which the myosin ingresses. Any relation between them? Our theory predicted that there is some sort of a universal curve. So the way we did in the theory is that we played with a bunch of parameters, aerodynamic length, bending rigidity, that, this, and so on. Also averaged over a huge number of random initial conditions, that gives you the error bars. Despite all that, there seems to be some nice clear curve that's predicting how the ingression rate of myosin depends on the accumulation rate of myosin depends on the ingression rate of the radius. We find something reasonably similar in the experiment. So the experiment is done by knocking off several genes, doing RNA perturbation of genes, and then redoing all these experiments, getting the 3D data, segmenting, analyzing, finding out the ingression rates, accumulation rates, and then asking how do the accumulation rates depend on the ingression. So the theory of active deformable surfaces seems to quantitatively capture both the kinetics of ingression and the myosin accumulation in the ring. Now the asymmetry. So this is the symmetric ingression that I showed you earlier. So you see the transverse plane and you see the two halves are ingressing symmetrically. If I just increase the contractility, you find that the ingression automatically becomes asymmetric. That's all we needed to do. It sort of makes sense. If you're at the onset of the ring-like patterns, so the flows are slow, and so therefore you have enough time for diffusion, turnover, bending elasticity, things to isotropize and give you something which is symmetric. If you're doing it very fast, you're not giving it enough time to relax. So therefore any anisotropies, any inhomogeneities will get amplified. And that's what gives rise to the asymmetric ingression. But not just that, we want to do quantitative measurements. Our theory predicted that the asymmetry, which is a measure of which is the displacement of the ring compared to its original position, that increases exponentially with time. We found that true in the experiments as well. There's also a rate constant, which is k delta, associated with the exponential increase. We can do the same measurements in the experiment as well and ask the same question. Is there a relationship between the geometry, the radius constriction, and the asymmetry increase? And surprise, surprise, both theory and experiment seem to predict a master curve on which both of them lie. Remember, we didn't put in any initial conditions favoring a ring or radius going down linearly. All this just came from geometry being solved consistently. Okay, so not just the fact that there is a cell division, but even asymmetries seem to come out very, very natural. So this is the phase diagram. So in a different plane, there's the contractility and some length scale associated viscosity and friction, homogeneous patterns, spot-like patterns, ring-like patterns. And at the onset of the ring-like patterns, you get patterns which are very symmetric. As you increase the contractility, the symmetry breaks and you get more and more asymmetric ingression. Okay, I think that's it. I don't know if I have one more slide. No, I don't. Okay, so I wanted to stop there and show you and wanted to summarize that if you include mechanics and geometry in some nice fashion, fun patterns seem to come up. Now, this is cell division where we are shown quantitatively. Surely you should be thinking cell division is the only case where there are dynamical geometries. No, all kinds of tissue deformations that you can think about. Invaginations, folding, looping. If you have a mechanism for trying to understand how geometry changes dynamically and how the geometry feeds back to the patterns, you have a way of, you tell me the geometry you want, one can try to construct that. So one would like to couple to realistic system where obviously there'll be signaling and controlled by genetic programs and all. We'd like to see how signals from genetic programs deform things and give you shapes and 3D functional forms. That's a long time goal that I would like to achieve. All right, thank you. I'll stop there. Yeah, if you have questions, you can bug me.
Yes. So when you said membrane that you are also solving, so are you also solving an elasticity problem coupled with the fluid there? Okay.
Elasticity for the bending, only for the bending.
So you have also a FSI interaction as well.
What is FSI?
Fluid structure interaction as well.
Like I told you, there's no cytoplasm. We've neglected the cytoplasm. There's only a deforming surface. There's a surface, there's a fluid there, and it's elastic for out-of-plane deformation. This is classical bending elasticity.
Okay. Okay.
Curvature elasticity.
Thank you. Yes.
Okay.
I had a quick question. So in the regime where, let's say, after where you show this linear velocity decrease, the velocity is linear.
Linearly, not the velocity.
Rather,
radius is decreasing linear. Yeah.
Constant, right?
Correct.
So is that something that you can
Analytically predict?
Is there a
Yeah.
okay.
In the paper, you'll actually find we have an argument to show if you go to the regime where the neck is just about forming,
Okay.
you can do an approximate calculation and analytically predict they should go down linearly with time.
Okay.
And also the fact that the myosin should go up exponentially with time. Both of this we can predict analytically.
Okay.
Asymmetry we cannot.
Okay.
Because asymmetry, I mean, by definition, it does not have azimuthal symmetry. There we can't do anything analytically.
Yeah, I have a question. Yeah. So will this sort of work if I don't have a single cell dividing, but like a collection of cells which maybe...
No, not there yet. This is just one surface deforming. That itself. Not.
Yeah, I have one question. So maybe it was clear. The surface, is it fluid or is it fluid with some elasticity?
So this is a classic membrane problem. So the lipid bilayer membrane of the cell is a 2D in-plane fluid, but it behaves like an elastic material for out-of-plane deformations. So remember, I showed you the membrane forms because you have lipids which have hydrophilic, hydrophobic interactions. It's a self-assembly problem. So the lipids diffuse freely in the plane of the membrane, so you cannot retain shear stresses in the plane of the membrane, but they do resist out-of-plane bending. So the elasticity is only for the bending part, and the in-plane is completely a fluid.
And there is a cortex below the membrane? Or?
So I'm thinking of both the membrane and the cortex as a single surface. I don't distinguish.
And everything is fluid?
Yeah,
Yes.
it's a one surface membrane composite. Yes. Yeah.
Yeah, so this is just a small question, but so the friction that you assume, it's just gamma V, I
Correct.
guess.
Correct.
So how good of an approximation that is in a real physical system?
Yeah. So, I mean, like I showed you earlier, the real thing would be to actually solve for the cytoplasmic flows inside and then use the traction to put it there. If you try to do that, you'll find, like I said, there's a length scale, which is the shear viscosity of the surface fluid to the shear viscosity of the cytoplasmic fluid. If that length scale is, I don't remember, very large or very small, you can show the tractional, full traction force will essentially be like the gamma V. This is like a thin film approximation that you're doing there. Remember, it's hard in the cell to maintain coherent flows in the cytoplasm. There are lots of other stuff. There's the nucleus then. So it's very, very unlikely that there is large scale crosstalk between this part of the cell surface and that part of the cell. It must be only on the surface that's going on. But that is a caveat. It's also technically much, much harder to solve cytoplasmic flows inside a surface which is deforming with time. Because I have to solve a PDE inside, the boundary conditions for that comes from a PDE on the surface. I don't know how to do that yet.